right, so a little overview about what we're going to talk about. Just a couple of slides sort of reviewing where we are, put this into context. Then I'm going to talk about how to calculate the allele and genotype frequencies. We'll spend some time plowing through the concept of probability, and we'll talk hypotheses and likelihood and interpreting the test results. And then finally, we'll conclude with a little bit about the assumptions that go into calculating match statistics. I definitely recommend you look at your Appendix 3 and certainly Chapter 11 in your book. Butler does an excellent job, of course, of presenting the information. So I'm adding some context and building on what he's written. I'm not trying to repeat what he's telling you, right? Okay, so off we go. Just as a reminder, it's not that I'm trying to sell my book. First of all, it's free, so that doesn't really do anything, but it is obviously provided free for a reason, as a resource for you to use. One of the things is that when you go to graduate school, you buy a bunch of books to use, and the only real book that I use anymore is my statistics book, which is kind of ironic. But what's happened with the internet, of course, is that there's now tons and tons of resources out there for you to learn different concepts, including statistics. So there's many places to go, but a nice tutorial site is at this web address. And then I have mentioned, and I'm just going to remind you, that the software programming language R is now certainly forensics using this more and more, and so there's a nice free book called Open Forensic Science in R. So just want to let you know about those resources. Okay, so our question is, how often does a particular allele show up in a or the population, right? So first off, what do you mean by population? And so from the genetic perspective, of course, we're thinking about a person is diploid, and so therefore at each position in the nuclear genome, there's going to be 2 copies of that gene, right? Or that position or that locus. So, but of course, population of individuals, then there can be far more than just 2 copies of that gene circulating in the population. And of course, the frequency of alleles in the population are the result of inheritance, parents to offspring, etc., etc. All right, and is also part of the history of that population. So alleles may have never been present in the population in historical time, but may be introduced through migration or immigration, right? Okay, so history matters as well. So considering our short tandem repeat alleles and using CODIS, okay, one of the considerations for doing certain kinds of analysis is who's actually in the database. Are they random samples drawn from the population, or are they collections of individuals who have come into the database for other reasons? We won't talk about that quite a bit. Your book has a paragraph about that. So stay tuned. There's a little bit more to that. I'll conclude with a summary slide. So we introduced back in the I think in part 2, if we want to know how many alleles of a particular type are in the population, the natural choice would be we'll go count them all, and so do a census. You should recognize that's impossible to do, and so we're going to have to do some sort of sample from the population. So an enormous effort has to go into thinking about how representative the sample is, so the quality of the sample. So your gold standard is do some sort of random selection where every individual in the population has a 1 divided by n chance of being selected. If you have that, then your results can be generalizable to the population. Okay? You don't have a degree of uncertainty associated with your counts. Once you have to do selection, though, and you realize that random samples have to be extremely large in order to recognize every minority group in the population, so probably going to have to do a modified random sample, and so that gets into a little bit more details about how to particularly do that. So the conclusion is sampling is kind of hard to do, and there's a number of non-trivial and non-obvious ways that sampling can be biased. So I thought I'd just give one example, and then we're going to move on to calculating allele frequencies. So one kind of bias that is very hard to overcome is sort of the rule that, well, the more you look for a particular thing, the more you're going to find. And that doesn't necessarily mean that that's equal to the base rate, but so anyway, it's called detection bias. So as an example, when I was driving in or writing these slides, I was sort of reflecting back. So how many VW buses did I see on my drive in? And the answer is I didn't remember any. Okay. And so then the next day, basically, I purposely looked for vans, and I saw 2. One of them was parked in a driveway that, by the way, I had passed the day before, but I just didn't notice it, right, or didn't think about it. So detection bias is an important source of error in designing samples and how you go about observing things. And so I'll show you how this affects our probability calculations in an example down a few slides from now. All right. So let's turn to box 11, and we'll use that as how to do our allele counts, right? And so obviously the author has already provided you the counts, so I think let's make a spreadsheet and show you how to do those calculations from scratch, because of course there's a lot more alleles we can give you, and so you should be able to do that at minimum. So the rules for making a table entry is to consider that there's 2 alleles in the population, say A1 and A2, and so the frequency of the A1 is simply, we'll give that P, and then the frequency or the probability of A2 will be Q, and so of course P plus Q is equal to 1. For the genotypes, then you square that binomial, right? P squared 2PQ, those are the heterozygotes. Q squared, that's the homozygote A2. This is P squared is the homozygous A1. And again, if you sum up those frequencies, it's going to equal 1. All right. If we want to then, if we're given genotypes, how do we get the allele frequencies? Well, you recognize that there's 2 copies in the homozygote, and then there's 1 copy in the heterozygote. So we're going to use this rationale to work from the box 11.1 to pull out our allele frequencies from the genotype counts. Now I'm just going to say, without sort of elaborating, this is just a binomial, but obviously the CODIS alleles are more than just 2, right? And so extending this into more than 2 alleles is just an accounting process, but instead of having P and Q, you're going to have a frequency for each one of the alleles, and so therefore you're going to have multiple heterozygotes in there, and this equation gets quite large depending on the number of alleles that are there. But the principle is the same. So if you understand for 2, you understand then for more than 2. Okay? All right, so off we go. So this is a representation of a spreadsheet. Okay, so columns A through L, rows 1 through 11. Your box 11, the sum of the allele counts is actually in column K. I've moved it over here because my head's blocking. So, okay, so at any rate, I don't know if you use spreadsheets very much, but they're really good ways of manipulating data and managing data. And so certainly for working with hundreds and hundreds of samples, it's reasonable to do this still in a spreadsheet. Once you get into thousands and tens of thousands, you're going to have to set up a database to do that, but the principles are the same. All right, so let's, the example in the book is describing how to get the allele frequency for 8, right? So as a reminder how to read the table, so this first row, those are the alleles, and in this first column, those are the same alleles again. I've only shown a few rows, but this goes 8, 9, 10, all the way down to 15 again, right? And then within the cells, those are the how often the genotype was observed. So to get the number of 8 alleles, this is the p squared, the homozygote, so we're going to count 9 twice. And then we get the alleles from the rest of the cells on that row. So come over to K and type in equals, and then the equation that you're going to use is sum, parentheses, 2 times B3, so that's how I get my 2 copies there, comma, and then C2 colon I2, so that gives me the sum of all of those cells, right? And then I hit enter, and boom, the spreadsheet does the addition for me. Let's get the allele frequency. So this is K2, so we just type in L2 equals K2 divided by G11. In your case, probably you'd set the 604 to be on K11. Just a real quick point about the $sign in front of the G and the $sign in front of the 11. What that is called an absolute reference to the cell. Without the $signs, then it's a relative reference. And so relative reference is really handy. So it's like once I set up this equation, I could just drag it down and it'll update the row numbers for me. But I don't want it to do that. I want to always refer to the 604. And so each time I calculate the observed frequency, if I drag this down, it won't update the 11. It'll maintain that. So that's a fairly nice little trick. You can ignore it, but that means you have to type it in all the time. All right, so this confirms that we can do the calculation for allele 8. So let's go to allele 9. Okay, so where do we find allele 9? Well, we find it in row 3, and the homozygous is C3. So when we do our equation again, equals 2 times C3, so that gets those guys. But notice we also find allele 9 in C2. It was genotype 8, 9. So we need to count those. So I just go ahead and grab that cell, and then another comma, and then I do my range again. So D3 through I3, and we got them all. Hit return, and boom, confirms the number from your book, right? Update, get the observed frequency for the allele, so that's going to be K3 divided by 604 again, right? 604 is the number of chromosomes. The sample size was 302 individuals. Okay, so let's one more example. Let's do for 10. All right, so where do we find allele 10? Row 4. And then we're also going to find it in D2 and D3 as part of genotype 8, 10, and 9, 10. Okay, so we add our formula, 2 times D4, that's the homozygous, and then D2 through D3, that's those 2 cells, and then finally the range E4 through I4, and 31 we get. Update the allele frequency, and so on for the rest of the alleles. Okay, so as a reminder, statistics describing the middle, describing the variability of your samples. That was described in Chapter 3. That was also part of Part 2 of our series here. And so description also includes, sometimes pictures are really useful. So Chapter 4 in my book covers graphics, and so I thought I would throw just a couple of examples in here in relation to allele frequencies. All right, so sometimes a picture is worth 1,000 words. So if I look at so I have an x variable and then 4 y variables, and the numbers for the y1, 2, 3, and 4, sort of look like they're all over the place. And in fact, if I calculate the mean and the standard deviation for y1, y2, y3, and y4, they're all the same. So if I just report the mean and standard deviations for y1, y2, y3, and y4, there wouldn't be much to say except for that they're equal. So this is why you also always want to generate graphs. Even if you don't report the graphs, you want to take a look at them. So a scatter plot would make a lot of sense here. Okay. There is my animations. Okay, so without going into a whole bunch, here's the horizontal axis is the x variable, and then the y-axis is each one of the 4 y variables. And what you should be able to see almost immediately is that the relationship between x and y1 is different from the association between x1 and y2, x and y4, x and y3. Right? Okay, so always do graphs. Even if you don't report them, it can give you a different picture to describe your results. All right. On the other hand, you shouldn't always think that you need to give me a graphic, just because we tend to think of ourselves as visual. So sometimes if you have just one number, just report it in a sentence. All right. So if the allele frequency is 11.2% and you're only talking about that one allele, a sentence will do. If you're making comparisons, sometimes a table is better than a graph. And so this is for jobs that are talking about bioinformatics or biostatistics, use of ArcGIS, say statistician. And so a simple table gives me the comparison very easy. You can imagine, of course, well, gee, why don't I put that into a graphic? And it's like, well, because it's kind of overkill, right? All I want to show is that statistician is a very generic term, and it shows up in hundreds of jobs. Compared to bioinformatics, nobody used that back in 2017. All right. So when you do graphs, these are the common choices. For allele frequencies, basically bar charts and pie charts are probably going to be the majority of your choice, but I thought I would just throw in some of the other types that you're going to commonly come into contact with. We just demonstrated scatter plots to show relationship between 2 ratio scale variables. And then, of course, there's a ton of specialty graphics. And I'm advocating strongly for those of you who are visual artists, please get involved in data visualization. We need all of the help we can get. It's really hard to do graphics that communicate in less than 1,000 words. Okay, so here's our allele frequency options. And so what I'm trying to sell you is that if you're thinking pie charts, please don't do pie charts, do bar graphs instead. So this is the same information, and notice that in order so we can help see things, we start color coding. Okay, I'd have to add labels in here so you could tell which allele is from. And also, you see this tiny little sliver here? That's the one allele 15 that was in that sample of 302 individuals. And so here it is right there in the bar. So bar charts are a better choice than when you're counting compared to pie charts. And just to sort of drive that point home, what if you're comparing 2 things? So like, for example, you want to show the relationship between 2 alleles. Pie chart, you're going to have to do some sort of exploding pie in order to represent those different frequencies, whereas either a table or a bar chart is going to give the comparison a visual that's far easier to look at. All right. Okay, so stand up a little bit, take a little break. So we're going to take you through discussion about probability. And so as our example, the allele frequency for the 8th allele of that D13S317 STR is 11.3% in that sample in box 11.1. Okay, so probability is simply the chance that an event will occur. It ranges from 0 to 100%. Okay, so what do you mean by an event? Well, for example, if I randomly pick somebody from the population, what's my expectation that they will have at least one allele 8? And the answer is 11.3% of the time. Okay, will I win the lotto? That's an event if I win the lotto. We don't have the lotto in Hawaii, so what's the probability that I'm going to win the lotto? 0. Okay, A1C levels below 6% is considered healthy. Above that is considered you're pre-diabetic. And all right, so what's the chance that, okay, so we can figure that out by doing a random sample of people and counting up the number of people who are below A1C level of 6%. Positive COVID-19 test. The positivity rate is still non-zero 2+ years into the pandemic. Okay. Positive drug test. So when you go to get a job, you're going to get tested. And what's the chance that your test result's going to come back positive? And if you say it's 0%, I have news for you. It's not 0%. So even if you don't use drugs, stay tuned. And then finally, just as my example, what's the chance I will see a classic VW van on my drive home today? So those are events, right? Okay. Probability, though, is usually taught from the perspective of theoretical probability, right? You know every possible outcome based on some theory. Hardy-Weinberg rules, as discussed in your book, Mendelian inheritance, monohybrid, dihybrid crosses, independent assortment. Those rules allow you to calculate with precision how many alleles you would expect to find in a population or in a F2 offspring of a monohybrid cross. Okay. Far more likely, though, particularly in biology, we don't have any theory to guide us. So what we have is, of course, 100 years plus of research covering a variety of things that interest us. And so we can derive our statistics, the likelihood that an event happens, simply by looking back at our studies. Then also, you should consider that some probabilities are objective. In other words, independent of the observer, different people are going to come up with the same probability estimates, as opposed to subjective probability. And so this one in particular is of interest in the legal profession, because obviously the prosecution believes they have the right person, and the defense is saying that you don't have the right person. And so this is an example of subjective probability, a reference to one's own experience or belief in a precept. What's the likelihood of this happening? Okay. This is my favorite one. All right, so it's going to take a little while to set this up. So Bob's 24 years old. He's applying for a job as a forklift driver with the widget company. Human Resources informs Bob he has to be tested for illegal drugs. So he goes to the lab immediately, doesn't stop anywhere, and he fills out the questionnaire and of course checks no to use of any illegal drug. And he also checks no when prompted to report any prescription use of opioids. Remember, medical use is legal, although if you test positive, it's going to generate some HR issues for you, but as it should, right? Okay, so results of the test. Bob tested positive. Bob's not going to get the job, so is that the end of the story? Right? Is Bob an opioid user? Well, he tested positive. So is that the end of the story? Let's see if you can get this. All right.